Hello, this is Miss Dagenford, and this should be a nice short video that will just get us started going tomorrow. And this is about active transport. Now, again, this should be a bit of a review for most of you. You might recall this back from biology or anatomy days. By the end of this video, you should have a pretty good sense of the direction the molecules move, what is needed for that to happen, and some examples. So active transport, and I have, concentrate, because that's what it does. You are going to be going in a direction that is from an area of low concentration to an area of high concentration. And so remember, that's directly opposed to diffusion. Diffusion, you start with a concentrated area and then it spreads. Well, this is the opposite. You're gonna try and get into a high concentration. Now, because of that, this is not passive. It takes energy. Now, it's gonna take energy in the form of ATP. Now, we haven't gone over ATP specifically, but ATP, if you kind of dig back in your mind a little bit, ATP is our adenosine triphosphate. And adenosine triphosphate is a molecule that the cell uses as little energy packets. And you might recall that the mitochondria and the chloroplasts are both kind of purveyors. They kind of produce ATP. They convert it. And so ATP is going to be our little energy source for these. And what they're going to be used for is changing the shape of proteins. And that's going to help shift the molecules from one side of the cell to the other. Now, most of these molecules can't cross the membrane anyway, or if they can, we want it to go in the opposite direction. So the best example, a very classic example, is the sodium-potassium pump. And the sodium-potassium pump, you find it in multiples of cells. Lots of examples of cells would have this. By far, the best example would be neurons. Neurons are a really good example of cells that would have an amazing number of these things. And the idea behind the sodium-potassium pump, it's going to be shaped in a way that can shift 3 sodium outside the cell and 2 potassium in. And because this happens over and over and over, that's what happens. You're going to start stockpiling sodium on the outside of the cell, and you're going to start stockpiling potassium on the inside. Now, the way these proteins work, so here's our purple protein. And this protein has these, this is a model of the protein, and this model is indicating that it's going to hold a very specific shape molecule. In this case, it's grabbing onto sodium, so these little circles are sodium. And then here's where the ATP is needed. And anytime you see these little explosions, that's ATP. And so ATP is going to come, and notice in the next picture what's happening is that molecule then can shift its shape. And so the ATP is giving that energy to that molecule to just open up and kind of rock open to the other side. So it's kind of this little swivel of a protein where it swivels to the inside and then it shifts or shuttles the molecules on the outside. Now, once the sodium molecules are on the outside, that also then leaves these pockets for potassium. So potassium binds, and notice this is going to be still charged with the ATP. Down here, this is showing that the phosphate leaves, And when that happens, notice what happens to the shape. The shape rocks back to normal, which means it opens back to the inside of the cell. So this is the inside, this is the outside. And if this keeps happening, if this repeats, you're constantly chucking the sodium outside the cell and then constantly moving the potassium inside the cell. And so what would happen, let me just draw you a quick diagram. Here's our neuron. And the neuron would have its axon, this long extension. And just for the sake of clarity, neurons are going to be nerve cells. And so they're sending signals. So this is a real important part of sending signals. We're going to talk about that briefly on Friday. And so what's happening is all along this part of the neuron, this axon, you're getting the sodium pumped out, and you're also getting the potassium pumped into it. So if I were to use my colors to start representing it, the sodium starts building up outside, and then the potassium would start to build on the inside. Now that doesn't mean there's no sodium on the inside or no potassium on the outside. It just means there's a higher concentration. We have built the concentration up, and that's active transport. Another example of active transport is a proton pump. And a proton pump, just as the name implies, it pumps protons. And protons are hydrogen ions. So in this example, the low concentration is over to the left. And again, we're going to have ATP, which is going to drop off that little bundle of energy to our protein. And that protein is going to shuttle the positive charges across, and then it's going to pile up. Now, these would not be able to pass through the membrane, neither would the sodium or potassium. And so we're going to be able to pump these out, and we see these in plant cells a lot. We see these in the digestive system, in the stomach, for instance. And you're making this area more acidic. It would have a lower pH. And in fact, you can do experiments on these by manipulating the pH and manipulating the low and the high concentration, and you can see in what direction these move. So there's another good example. Now, just as a quick aside, here's a good example of where you might see the cell with a proton pump. If this is our intestinal cell, I'm sorry, not intestine, stomach cell, my bad. If this is a stomach cell, and so this is the inside of the stomach, and this is where we would want a low pH, maybe pH 2. Well, you want lots of protons. You want a lot of hydrogen ions, hydrochloric acid, and so you want to be pumping those protons out. Now, what's interesting is, let me give you a for instance. Let's say the membrane has all of these proton pumps all throughout the cell on all sides. Now, that's okay for the top, because the top is pumping protons to the inside of the stomach, which is where we want a low pH. But think about it. That means this is the outside of the stomach. If the protons are getting shunted to this, you would be damaging the stomach wall, the stomach lining on the outside. So what stomach cells do, they have kind of an interesting makeup. They can corral, and they're going to use other proteins, to kind of create a little belt that prevents the proteins. Remember, the membrane is fluid. And so normally these proton pumps would be drifting around the membrane, but cells can corral their proteins and make sure that they stay on one side of directionality. That's just kind of an interesting side note there, but there's an example of a functioning active transport protein. Now, so let's just do a quick comparison. Let me zoom in. So here's going to be a quick question to see if you can identify what A is referring to, what B, and B would be both of these, what B is referring to, and then what part C is referring to. So this is just, can you understand the diagram? Now, just when you think you got them all, here's going to be an interesting connecting point. Some cells, we do use this with our sodium-potassium pump. Plants use it with the proton pump. They use what's called co-transport, and they put active transport and facilitated diffusion together. So let's look at this diagram. Here we have our ATP going to this protein, which should give you the clue that it's active transport, and we've got a low concentration and a high concentration. So this is going to be our proton pump. Now, once we have our hydrogen ions, our positive charges, once we have them out here, this is now a high concentration of hydrogen ions. Once we have that, think about what that means. These molecules would tend to move back in. And they do. And they do through this co-transporter. And the co-transporter uses the drive of the protons diffusing in to also piggyback another molecule. So notice this is a low concentration, and I hope you are starting to think about what this might be. We'll come at the end and answer. So this is a low concentration, and then over here we've got, it's not a very elaborate, but it's a high concentration of that green molecule. So this is a form of active transport down here because it's pumping this in from low to high, but notice the hydrogen ions are going from high to low. So this is a form of facilitated diffusion. So it's kind of an interesting combination. And the reason this works is because you've got this high concentration to piggyback the low concentration of that molecule. And plant cells do this with the proton pumps. We do it with the sodium-potassium pump. And we will talk about exocytosis and endocytosis tomorrow.